We're thrilled to give this webinar, and I hope everybody can hear us. I'm Bruno van der Meulen. I'm responsible for the Digitization and Document Delivery Department at KULeuven, and I'm joined by Hendrik Hameel, our imaging specialist. Today we will talk about our standard multispectral imaging service and multi-light reflectance imaging. I will concentrate on the multispectral imaging, and Hendrik will expand on the multi-light reflectance imaging. We'll shortly touch the technology used, our roadmap, present some examples, and then have a look how we incorporate it in our library services. The digitization department has three focus points, one of which is research and policy development. It's part of the digitization roadmap at the KULeuven, and in this the imaging lab plays an important part. For our normal digitization, we focus on project management and our imaging lab. We have a focus on quality, but more important for this presentation, we want to highlight our collaboration with research, implementing techniques to advance research, development, and implementation of new tools. On the multispectral imaging, a few slides on technology. I won't go too deep into this as this is fairly basic. Multispectral imaging is beyond visible spectrum in the UV and infrared area. Important to note that the capture procedure is more complex than normal imaging. It's slower, you have more issues on storage, and you have extra avenues for research and dissemination. We use, it's camera based. We don't use a normal camera, but a modified camera, which is UV and infrared sensitive. You can use a black and white camera without a biofilter, or you can use a modified color camera where the infrared and UV filter has been removed. You also need a lens, a good lens. Not all lenses are suited for multispectral imaging, as some lenses have hotspots, as you see on the right. Also important to notice is that there's a shift in plane of focus. The point of focus in the infrared spectrum is different than in the visual spectrum. So for this you have special lenses, specialty lenses, full apochromatic lenses with fluoride glass. So when you focus in the visual spectrum, your image is also sharp in the infrared spectrum. On the light, you have two options. Either emitted light is broad spectrum or narrow spectrum. If you have a broad spectrum light, you filter out the light you need. So you put a lot of light on the object and you only filter out what you need. On the other hand, when you emit light in a narrow spectrum, you only emit the light you need in the spectrum you need. So this is more accurate, but it's more expensive and less flexible. Filters are more flexible than lights. What we found out is that calibration is also really important. In conventional photography, you have established standards such as Metamorphose or Fudgy, and you have commercially available calibration targets such as a color checker. But for multispectral imaging, there is no standard, or there is no standard calibration routine. There are a few options. such as the UV target by UV innovations. You have the pigment chart. We will focus on that later. And you have diffuse reflectance standards such as Spectralon. Doing multispectral capture produces a lot of images of one and the same object. So this is already interesting, as you can see difference between the different spectra. But with the processing, you can do more. You can visualize features hardly or not visible to the human eye through false color images, enhancement processing, or even more advanced processing routines such as PCA or spectral unbinding. We will present now our roadmap we developed over the past 10 years and look at some examples. We started about 10 years ago with a small experiment. We had a question from a researcher. He could look at this ostracon, and it was barely readable, and he asked me if I could enhance readability. The older camera, Nikon D100, had limited sensitivity in infrared region, and I acquired cheap infrared filter online, and the researcher was really happy as he could at least read one sentence that was not readable before. So this was really our first step into multispectral imaging. In 2011, phase 1 came out with black and white back with enhanced sensitivity in UV and infrared. We were able to test it for 1 week with a standard lens and also this infrared filter, but we also tested with acetate filters. And this was really interesting to see. We got some results showing some underdrawings in manuscripts, and also in the UV spectrum we used the black light, showing the topography much better than with the normal visual light or the infrared light. So this was really more an experiment than establishing a service. Over the course of years, we slowly invested in material, in infrastructure, such as a filter kit. We experimented with forensic photo kits by Pekka. On the right hand, you see some graphs of spectra, of transmissive spectra of the filters. And we also bought additional filters, really narrow bandpass filters of 10 nanometers, so we could pinpoint exactly our spectrum used. First assignment was this antiphonarium from private collection, where we developed a simple capture procedure, where we also had immediate results showing the underdrawings and also the surface characteristics of the illuminations with the UV. So for the researcher, for conservators, this was already additional information they could use. On the light source, we used a normal flash, standard studio flash for the infrared and visual capture. And with the UV, we used a special UV tube or a black light. In 2012, we acquired a grant for development of a multispectral portable light dome. Hendrik will focus on that after the multispectral imaging. And with the EES project on ancient Egyptian figurines, we established a simple standardized recording and processing pipeline. For this, we invested through the funding in a full spectrum converted Nikon camera. We already acquired before our apochromatic lens, and we started experimenting with processing. So here you see the type of material we have been working on, and this showed us the importance of post-processing. While the multispectral captures individually already give you interesting information, it's by combining those images and further processing that you can get more information out of it. And on the right hand side you can see a simple Photoshop mixing of color channels and extraction of information. Last year we invested in better filters, in more filters. So we have a set of 14 filters, narrow band pass filters. It was a selection we made, so we had an overlap of the entire spectrum between UV and infrared. Before we worked with only 5 filters, with a 10 nanometer narrow bandpass filter in, as you see on the image on the left, with a filter wheel, a manual filter wheel. Unfortunately, a filter wheel of 14 filters doesn't exist, so we had to look to other solution, and we equipped our filters with a magnetic and our lens with a magnetic filter ring, so we could quickly change our filters while doing our capture procedure. Here you see different spectra, and on the image on the right you see us at work with the Codex Arkansas, which is the oldest known manuscript produced in the Low Countries. New filters also meant that we wanted to invest time in new calibration routine, as we experienced that the normal calibration targets were not suitable for UV or infrared imaging. We were happy to borrow a spectral on target from colleagues here at KULeuven University for testing, and what you will see now starting is animated GIF passing through all spectra, and you will see that in the target below the infrared will get darker and the UV will also get darker. So using a normal target based on Munsell patches for calibration is not really good beyond the visual spectrum. With the spectral on target on the right, you can calibrate your capture procedure much better, also in the UV and infrared. And with the spectral it stays consistent throughout, , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , , So we invested in a spectral target, and as you see with the graph, it stays flat. There is no peak in the infrared or UV spectrum. So when you make a capture with a spectral target, either in UV visual or infrared light, it will show up as light or as dark as throughout the capture. So what are the limits of standardized multispectral capture? It's quite time consuming. It takes easily a few minutes per sequence. As you want to process the images further on, you need precise registration, so stability of the capture routine is really an issue. For the post-processing, the spectral on target allowed us to fine-tune the reflectance, and you really need a dark room as a good quality capture. What we have shown here, we have summed up everything, and we come at a price of about 13,000 euro, which is not cheap. But on the other hand, using filters and camera, it allows you to spread your investment over time. And as we did, we built this up over the course of years. For the standard output, it's for conservation, looking at underdrawings, looking at the materiality of objects. It's a relatively simple procedure. It's doable for a normal institute lab. For us, it has become a standard service. We can incorporate it in our service catalog, and it's really meant toward research and conservation. And now we will show an example of an extra service we developed. We start with the capture of reference material. This is the historical pigment checker by Cultural Heritage Science Open Source. We capture the whole routine with all the filters. Next, we capture our heritage object with exactly the same settings. And we can use this capture routine for identification of materials. Since we have captured our reference material, and this is the historical pigment, we can extract metric data from our captures through ImageJ. It's open source image processing software. We can extract this metric data and we can compare it to other analytical data. And this method was proposed at Archiving 2018 workshop. And here you see a final result. We have in black the spectrum measured through our capture routine, and next to that spectral fingerprint made by an XRF. It's more analytical data, and you can compare them and see that they match. And in this case, we were fairly certain that the blue pigment was azurite. You can do this for every pigment, but there are a few set pigments that you can assess this way. So what is our roadmap for the near future? We have acquired funding for a new project to acquire new equipment. It's a 3PI project: papyrus, parchment, paper, and imaging. It's a collaboration between Book Heritage Lab of Livo Illuminare, ESAT, and KULeuven nowadays. We want to integrate both visualization and analytical tools, and tools for data creation. We want to establish more tools for processing and have a solution for dissemination of multispectral data. And I'm happy to pass on the word to Hendrik Hamel.
So, good morning to everyone. I hope you can hear me well. I will keep the chat in my left eye to see whether the sound is okay. I will continue this talk with what we have been doing with multi-light reflectance imaging. Both the technology and the roadmap I will approach at the same time. Sorry for this, something wants to update on our computer right now. Okay, thank you. Obviously, when we are talking about this system, we are very much aware that this system is not a system which can be used in a standard imaging lab. This is something we have developed together with our university department of engineering. It has started already in 2003. It has been part of multiple funded research funds. And around 2011, we also started using it for the digitization, so the advanced imaging of cultural heritage in our library. So we are very much aware that this is not a system which is easily to use, easily to incorporate in any standard imaging lab or digitization process in any kind of institution. This is something we have specialized ourselves in, but we nevertheless would like to take the opportunity to present this system and some of the results we have acquired with this approach, because at most conferences around the world, more and more this approach of multi-light reflectance imaging is being brought forward as one of the approaches in the future which will be used by many people involved in the heritage sector. So you can see here on this slide some important steps we have made. When we talk about that system, we have developed what we have called the portable light dome. And over the course of these last 15 years, when the development took place, and it's still going on, we have developed a number of different devices. On this slide, you can see a few of them. So some look futuristic, others already start looking archaic, but they all have their special abilities. We have two sizes, one we call microdome, the other one we call mini dome. Microdome is smaller than the mini dome, and we have also used different materials to make these domes. At the same time, with these domes, we have been using or introducing two lighting systems. We are using in these multi-reflectance domes, in one type of domes, normal white light, but then one series of these domes we have also equipped with several multispectral bands, that is ultraviolet, blue, green, red, and infrared. So then at the inside of these domes, different LEDs are mounted with different spectral characteristics, and as such, as we have seen in the first part of this presentation by Bruno, also many of the abilities which come from multispectral imaging can also be used on this multi-light approach. You can see here, these are all animated figures you will see right now, so probably they will not come through via this webinar as fluent as we see it ourselves, but every few seconds your screen is being updated, and so you will constantly see other images. You see on the left-hand side our viewer software we have developed, and so after we make a recording, all of the information is being processed and we can view the results, and you can see each time that when we ask different, it is up to us whether it is people from the imaging lab or whether there are people from conservation, whether are people from research. It is up to them now to start asking this interactive data, what do they want to see, what do they want to observe. And so in that viewer software, all of that can be done. What is the general idea, the general principles behind this system? Well, on the left-hand side, you see a schematic view of a dome-shaped device. In that dome-shaped device, you have many different light sources. They come from all different sides. So we start illuminating the object, which is positioned at the bottom of this dome-shaped device. And each time when one of the lights is being illuminated, one picture is being taken. And then in the middle, you see a large data set. Actually, you have to multiply these images by 228 or 260. So the microdome is 228 images, and the mini dome is 260 images which are being created. And based on such a data set, two specific characteristics of your surface are being established. First of all, it understands much better per pixel the reflectance of that specific point on your surface. And secondly, it also understands that specific point in what direction it is oriented. So that means that you actually also will start extracting three-dimensional information on your surface. And that kind of data, so the final compressed data file, once the processing has been done, and from the start of the recording to a finished final compressed data file, that takes something like maximum 10, 12, 13 minutes, and doesn't take much longer than that. So when you upload such a final processed file, you can start consulting it immediately, and you actually have a finished product. So most of the actions which have to take place during this registration and processing process can be done quite fastly by just clicking a few buttons. Based on these image data sets, for example, you can also start creating 3D models, which is an extra output which creates a lot of possibilities towards both imaging or dissemination of your data sets or further research. We will now have a look, and we will do it quite shortly, so there is enough time at the end of this presentation to answer some questions. So we will only have a look at a few results of this system. These are quite a lot of slides, but we will go quite fast through them. This is an example of a bookbinding where the lighting is coming from the top. So you can't actually see a lot of the relief on this leather bookbinding. But one of the first abilities of this system is that once you have such a data set and it is uploaded in your viewer environment, you can easily start changing the lighting angles. That is what you see on this slide or on the next one. So each time we are changing the lighting angles and different types of information can be viewed, all depending on from what light angle you have chosen to look at your object. In the middle, you can now also see two images, and on these images, we are now approaching that same visual data set, and we start analyzing the object with metrical data. So the green line in the middle of the images, each time they give you information on, so per millimeter, you can actually measure the surface, and with the blue line, you even see the profile, and so the actual topography of your surface. That is always incorporated in all of these recordings you take with the portable light dome. At the same time, with that same data set, and just by clicking another button in the viewer interface, you can also, for example, extract the color information, the texture, and then you will start approaching your imaged surface in a complete different way. Some of the topographical features, for example, will be much clearer. So again, a few images which demonstrate that. Or you can even start asking your software, as the data set conceals a lot of information on the topography, why don't you start processing that topography in a complete different manner? In this example, for example, we have asked accentuate everything where the direction of the surface is changing very rapidly. So pixel per pixel, but it happens in a millisecond, that calculation. It immediately accentuates the relief of your surface. So this is just one example. A second example of what you can do with these kind of imaging, advanced imaging systems, is, for example, when you start using it in the monitoring during conservation actions. Here we have an example of an illuminated manuscript and a detail on an illuminated manuscript. And we have imaged the illuminated manuscript before and after conservation treatment. First of all, we start using the relighting tool. So when we illuminate the object from different sides, you can immediately see that on the left-hand side and on the right-hand side, we have already a very good tool to visualize the conservation actions. And then without the color, it is even more obvious now. But then we can start using also, for example, this measure tool. We can measure the surface, are there any bumps in the surface or not. Before treatment, in this case, there were a lot of them. Well, after treatment, it has been flattened out, and as such, we can easily monitor, image, and also prove towards the rest of the heritage community that the actions have taken place. We can even extract that information, that metric information, and we can, for example, plot it in a graph to fully explore and fully present the results of such actions. And all of that is actually based on a imaging tool. A third example to explore the possibilities of this tool. Here we have a detail on the Bible of Anjou, on folio number 1, that is. And we have used the same tool, and as we are using for the multispectral dome 5 different spectral bands, you can also start understanding the spectral response of each of the materials you have been imaging, in this case, different pigments. On the left-hand side, you see that in a normal RGB image, we have a blue field in this illumination. But when we start combining the different spectral outputs, that is the image in the middle of the Bible of Anjou, actually that blue pigment or that blue field, it turned out to be actually 2 different pigments. And for each of these pigments, you can create a spectral fingerprint. And we use histograms in the portable light dome system to do that. And then again, as has already been explained in the parts that Bruno presented on multispectral imaging, we can use a pigment checker. Historical pigments have also been imaged with the multispectral portable light dome. And when you start comparing, this gave us the possibility not only to demonstrate and prove that with this imaging tool we can differentiate different pigments, but we could even start identifying the pigments by their reflectance fingerprint. A fourth example is on one of the, it is part of a large project with the name Fingerprint, which is on the graphic work of Breugel. This is a project which is still going on between KULeuven and the Royal Library in Brussels. And we have scanned some 200 graphic drawings and prints by Breugel. And we also have scanned a few of the original drawings by Peter Breugel the Elder himself. Which have been used then to make prints for the copper plate. And when we started using the multispectral portable light dome on that, we were even able to visualize and to capture the information on the tracing lines which were left behind in the process towards the print on the original drawing. Even more interestingly, as we use multispectral information, these tracing lines, for example, when we use the infrared data sets of our portable light dome, well, they were not that appropriate or in a good place to track these tracing lines. But if we used the ultraviolet results, these tracing lines were enormously easy to visualize. Then a final example of where we have been using the multispectral portable light dome in our imaging practices. Here, for example, you see in image number A that this is a normal picture of a print by Bruegel, which is a standard picture according to the metamorphose and Faggi requirements. And actually on this image, it is not that easy to see which zones have been damaged, whether there are zones on the surface with conservation treatments. For that kind of reasons, normally that is image number B, you would use, for example, a backlight photograph. But actually, when we wouldn't have the possibility to create such a backlight images with the portable light dome, these damages, and these are images C, D, E, and F, these damages which pop up very clearly on the backlight, they can also be studied with an enormous detail on the portable light dome images. How all of this, that is both the multispectral imaging already explained by Bruno, and also how the advanced imaging by using the portable light dome is being incorporated in the library services. And we will end with that, only a few more slides, and Bruno will go into this very shortly.
So we have our normal service with all our steps from preparation to post-care, ingest and publishing and post-care. And the advanced imaging, we place it in the enrichment phase of the of our service. First, you need standard good quality digitization as a reference. And on top of that, you can work on advanced imaging. For this, we have our imaging lab. We have a data work environment. We have a digital asset management system to keep track of all the data and keep track of all the derivative data, as we produce a lot of images of one of the same object, not only raw images, but also processed images. And we use this digital asset management system also for data delivery. And data delivery, this is delivery to our partners, such the research community within KULeuven, Book Heritage Lab, curators, and so on. As you see, there is no direct link between our advanced imaging and our standard procedure, our standard routine. So this is now something we will work on the coming years to include this in our normal processing and ingest routine. So up to now, this has been more or less separate. And the next few years, we'll focus on full integration within our system, within our preservation system of our normal standardization data. So what is the added value? First of all, you can develop an integrated service with both standardization and imaging, and in that sense, it makes both even stronger. and it's at the service of researchers. You can develop routines, you can develop workflows in close collaboration. Also with processing routine, you do this in collaboration, because some researchers are interested in zone A or technique A, other researchers are interested in another zone or another issue. As such, the advanced image is embedded on institutional level and not within research groups, where you have a lot of turnaround of research assistants, and when a research assistant goes away, his expertise also goes away. But by embedding this advanced imaging service within our imaging lab and within the university library, we can build up we can build up our expertise and give it as a service to researchers. It also allows us to build up high-end infrastructure and non-standard infrastructure through external funding, as we closely collaborate with researchers on proposals. Additional added value is this equipment is pooled and is not dependent less dependent on incidental research funding. It's open to more than one researcher, so this was also a good argument. We are embedded in the library, and the library is our lab to test out routines, to develop new workflows, to see if routines work out. And for the library, it gives us a higher visibility, not only in Leuven, but also in Belgium and even wider. We have come to the end of our presentation. We would like to thank you for your interest, and are happy to answer questions if those arise. That's a good question. How big are your data files? It depends on techniques used. With the microdome, we have a raw data set of about 5 to 7 gigabytes. All this data is kept and within our digital asset management system, but this is not our preservation system. And this is what I've been talking about. Our imaging service is separate from our standard digitization service, and the long-term storage and long-term preservation of these files is something we will tackle in the 3PI project. With the multispectral imaging, the standard multispectral imaging, the size depends a bit on the resolution of the camera and the images made, but it's also a few gigabytes per data set, including the processed data. So no, at this moment there's no long-term storage, but we will work on that now the coming years. Are the images online available? At this moment, not on a structural level. In a sense, it's research oriented, and these images are embedded in research projects, and as such are disseminated through publications and so. But at this moment, there's not an online data set accessible. But this has to do with two. I wouldn't say problem. Problem is not the right word, but two things you have to tackle. First of all, it's research oriented, and as long as it's not published, you can open it to the broader public. And on the other hand, with preservation comes also the opening up of the data, and we have a digital asset management system which works for us really well, but this is a closed environment and our preservation environment is open. So again, we will work on that later on. And when projects are finished, those projects, research projects, are not always KULeuven collections. We work a lot with external collections, and the opening up of this data is in collaboration with the external collection. So we can provide, we can facilitate that, but it's in close collaboration. Okay, thank you for the opportunity. And we really thrilled. This was the first time we did a webinar, so I hope everything was loud and clear. And thank you for your attention. Bye.
Goodbye.
Thanks very